So, so today I want to talk about ways that we might be able to improve drug trial. So today I want to talk about ways that we might be able to improve methods for testing out drugs, so we can both get safer drugs, get them more cheaper and on the market faster, to save many more lives. And I have three big ideas for that. They're kind of separate, but they can all work together. First idea is that for some drugs, we could reuse the placebo trial to some extent and the control trial. And this is how we would do it. Now, it's important that the placebo effect in a drug trial, there's three groups. There's a group that gets the drug, there's a group that gets a placebo, and a group that gets nothing. The placebo is important. What the placebo is a fake version of the drug. They try to make them think it works, or a fake version procedure, or whatever. Some things like pain, if you feel like you should feel less pain, oftentimes you do feel less pain. Placebo effect can be huge. It's even been shown to help in healing bones and injuries, because when you feel better, your immune system goes up, your general health, you reported side effects, and all these other things. And the placebo effect does vary from group to group, so you can't fully do away with a new placebo group. But however, what you could do is say that, hey, we had a similar drug trial happen all this time ago. Let's cut the placebo in half and use half data from previous drug trials, or certain things, only recent drug trials that had similar results. And we have ways to see if the placebo effects between these drug groups should be similar. So if the placebo between the two drug groups are not similar for the same disease for whatever reason, maybe times changed, maybe the formulation doesn't trick people as well, then we can increase the placebo effect size in a new one. Or we can pull the data together. But that means we can do away with a less big. But in cases that we can reduce dramatically the size of placebo trial, and we still want also a placebo trial, somewhat of a placebo trial, so we don't want everyone just to stop doing placebo trials and just waiting for one person to do the first one, because that means the initial investment is very high, and compared to other later ones. We can put more people in the actual, trying out the actual drug, which could mean that we get more data on the actual drug, which could mean for safer drugs. Or it could mean that more people think the placebo works, so the placebo effect might actually increase. So we get more reliable about the placebo one. The control group, how could we get the control group better? Well, if we had a national survey where we just inspect various drugs, conditions, not drugs, in much more detail, so that people have much better baseline data on these conditions before we do drugs, we could lower the amount of people we need in the control group, because we already have that data again. This would be very, very useful also ongoing forward, in that we could use this data to also understand this diseases in general better, so we could design better regimen or know how much of a problem it is, or who incurs more. We could also, to some extent, reuse control trial data from previous ones. These are all just basic ideas, and they would really dramatically reduce cost, but they won't get rid of cost altogether. But yeah, because it would also make the system a lot faster, because recruitment will come a lot faster. Recruitment will be a lot faster. Also, if people think they get the drug, they'll more likely sign up for a new trial. My second idea is to have more of a multiple level scheme for testing out new drugs. By what I mean by this is that we have multiple safety authorizations where we have really safe drugs, which we have a ton of data on, and then we have the less safe drugs. We already have this with over-the-counter versus non-over-the-counter, but I'm saying many more levels. And when we have these multiple levels, you're allowed to participate in them at a different level, but you must be part of the trial. Or we have multiple levels where we approve more dangerous drugs. And yes, I'm saying dangerous drugs, but we give a lot more warnings before using it, or dangerous procedures. So more stuff can get approved for the rare conditions where you have to give something dangerous, or that it's more experimental. So we can make approval easier, but put a lot of but ifs and have a lot of things about how it's more dangerous, a lot more warnings and safety guidelines, and say that you can't just stop designing it. And examples of this where safety gone away of new drugs is that we do have a procedure to cure actually multiple sclerosis. So multiple sclerosis is an awful disease, and this probably could work for almost any autoimmune disease, where the immune system will eventually kill you, but it starts attacking the myelin sheaths on nerve cells. So myelin is like an insulator around nerve cells that makes them transmit signals better. But the immune system in multiple sclerosis starts attacking it, causing you to slowly lose functions in your brain. Usually it's mostly muscles, then it affects memories and emotions, and it slowly goes down the list. We do have a way of actually curing the disease. That is, and it's been proven, and it makes perfect sense, is we destroy your whole immune system that's learned. So you have no more learned immune system, and then you get rid of the disease. Okay? Why isn't this allowed to use in the US? It's very hard to get. Well, there's a high chance that it kills the patient, because you have no immune system after this almost. You do have a tiny bit, but your immune system is weaker than a baby, and you have to relearn immunity to every single disease, and it's going to take you longer, and it's never going to be as strong as a normal immune system, because when you're older, you just can't learn the same as well as your baby to different diseases. So yes, it is a horrible treatment, but multiple sclerosis anyways is going to kill you. So it's worth the risk, and in some cases, and it can even reverse the neurodegeneration and regain neuronic function, because your body can't put myelin sheaths back onto it. And some more evidence of it being more than expected recovery. Another example of where this might be okay is male birth control. Is that male birth control, a lot of people have been avoiding male birth control, and they complain about the same side effects that appear in women to the same extent almost. But men don't have to worry about pregnancy and all these other things, so less willing to take it. But let's approve it. Tell them about all the risks and everything, and if some men want to take it, some women take it, because it's not fair just to have it for women, and for some men barely experience those side effects. This will also encourage people to try to find a better one as it becomes more normalized. Some men also barely experience the side effects, and some people in relationships, the men will experience less side effects than the women. So it'll be a good idea, and just for gender equality, but also putting the option out there, and for the people that have it safe. Multiple level approval should come with a stipulation that you should keep collecting data. We can give you a fast approval, but keep collecting data until we can get into a safe level, and you have to come up with a solid plan to collect data, and you can't stop trying to innovate and make new ideas out there, which I don't think will happen if there's good government funding that keeps pushing for new ideas. I don't think we can leave it just up to the private sector with this idea. The last idea I want to talk about, an idea that perhaps has the most math to it, well, definitely, but it's the hardest to conceptualize, is better stats. So nowadays what we usually use is, and I did a whole video lecture on this, which I'll link below, that goes much more in depth with much more cases, and you learn much more. So if you're really interested in this, I will watch the video. But basically what we use nowadays is stuff based on the bell curve or derivatives bell curves that are unimodal, and they only look at a simple distribution where you have a center. I'm drawing, everything varies around from the center, the same amount, it's symmetric, and that the center is and that there's a certain, goes up and down a certain amount, and we put all the drugs in one of these curves, and we put all the other ones in another one of these curves, and we see if it looks like the curves are different, or do they look like they could be on top of each other. The more they're on top of each other, the more similar they are. And this is a probability distribution. So the more they overlap, it's like two probabilities overlapping. Another way of visualizing how geometry can give two probabilities is if I have a graph and I'm going to draw time and that's ugly time location. If I draw my time and location in red, that's a point of everywhere where you could meet me. So this is everywhere you could meet me in red. It's all the different times and locations. I put them down. If your time and location is in blue, where they intersect, which I'll make purple, is where you actually do meet me. It's also, you can kind of think of like a Venn diagram. Venn diagram, where you have the two different intersections. We have my person and your person. Intersection is where the probabilities meet, and it chance that the two things happen at the same time. This method is very outdated because these curves are not necessarily unimodal. They can have multiple bumps, and I go over that much more in my other lecture and reasons why you would expect that. You would expect that if there's people with variations within the group that causes something to work a lot better for some people than other people, for whatever reasons, and that can be gender, that can be a bunch of other things, that can be race, that can be other health factors, diet and stuff like that might make the drug work very different for different people. Another one is that it just could be something you don't understand. This doesn't work if the distribution around the mean is not even. So you could have some people it works if it works for some people super excellent, but for other people barely works, this model does not work well. It does not work well in the opposite. So this model doesn't work well in a lot of cases. It also doesn't take into account something we call confounding factors. So confounding factors are factors that could mess up your data that are not it, such as the drug might not work as well if you smoke cigarettes at the same time, or they might not work well if you miss one dosage. It might have a huge impact or not. That's confounding factors. Confounding factors is anything else besides the drug that might mess up your data, or it either increases the drug's effect, decreases it, or just changes the symptomology regardless of the drug. And it might not, and sometimes confounding factors means that you might not have perfect two groups. Like randomly in one group you might have more women, and another group you might have more men. In one group you might have more people that are impoverished or have worse or good diets. And you might have some groups that have more intersections. So like one group you might have women and you have people that have bad diets, but another group you might have people you might have more women with bad diets, and the drug might not work as effectively in women and people with bad diets, and the two interact strongly together. So even though both groups have the same amount of women and bad diets separately, the person that has both of them is worse. So I would illustrate as that one group has one woman and one person with bad diet B, but the other group has someone that's both women and bad diet. Same amount, but it skews the data. So many ways the data can be skewed. So what's the alternative and to this model? That's random forests is one of the biggest one, but generally feature importance. So feature importance is the way you put in a ton of data, and then you make a model with that data. You figure out what features in that model are the most important. Drug, placebo control, that's one variable. You can have race, sex, gender, age, income, weight, a bunch of other things. And then you can separate out that and see what's most important. This way you can really see, does this drug or anything reduce to an impact beyond all these confounding variables. And the nice thing about it compared to these older models is the more confounding variables you put in it, the more accurate your model is and the better your model, and the more less likely it's to give you a false positive, which says a drug works when it doesn't work. It's less likely to overestimate, but also more likely to see small effects at the same time. Small effects when it might only be a minor improvement. And it can get rid of some of the things where you have compounding when you have effects of multiple things have effects together, like having poor diet and cigarettes increases your chance so much more than just either one alone. So that's basically what future importance is, but let's do future importance with random forest. So random forest is based on trees. This is a tree, what I drew here. Yeah, it's a mathematical tree. It's not a real tree. So we start at the base of the tree, which is upside down, as mathematicians do upside down trees often. And we have green and red dots. Green will say it works. Red will say it doesn't work. So then we look at variable A in our data. And variable A is the biggest thing in the data. And we just use variable A and we split the data using variable A. And we split it using a line that we fit with the data. And this line this is not a line, but we split with some curve. And that curve is based just on variable A, just one variable. And half the data goes to this side, half with more green, half more red goes to the other side. So that's the basic idea. Now, what is this? Now, in the second step, we look at what next one variable splits the data according to whatever model we're using, a linear split or splitting with a more complicated curve, and we find out that it's B, just randomly. Then we split it until these ones have more green, one side has more green and one side has more red, until there's only green or red or some other parameters at the end. And we do that for the other side too. Now on this side, we have more green and red. But by using a simple model, first, we look at the most important factor first, and we use that factor first in this tree. Then we look at factors that are less important, or we use the same factor. And because we use so many different tree branches, we can actually do it to all these complex models. Because the first model might be just a simple line split. The next model might be a split in that line that makes it look like this and this. So we get a more complicated curve. Then we might have splits like this or this. And in this data frame, we can have all these complex things because all these simple little models add up to a complex model within the tree. So that's the basic idea of a tree. And what the nice thing about a tree is by working way back up, we can see which variables are most important, how important they are, and how predictive they are. And we can just see if some data is just random. Now there's a way that we can make these trees even better. Trees by themselves are not very good, because trees tend to overfit, or they overestimate based on a little bit if they see all the data at once and make a too complex model. So how do we make the models, a bunch of small models, even simpler and more general? Well, we fit one tree to a little bit of the data, so tree 1. Then we fit another tree to a little bit of the data, tree 2 that's different, and so on and so on. So none of the trees see all the data, but it's a bunch of them, and we call that a forest. And then we can really do the same idea, but then we can really figure out which data is most important or not really quickly and pretty precisely in most cases. This is a lot more accurate than I can say, than almost any other method out there. And it also, for computers, it's easier to calculate more precise than the older tests. The older tests are actually better by hand, but with computers, this one becomes easier for a computer to calculate. Computer can calculate literally tens of thousands of data points with 10,000 variables. My Mac computer can do it in minutes. Fit this model and figure out this. So then, in a random forest, if we want to make it even better, a random forest, we use something else to boost it. And that is, we can test it only on 60% of the data, 90% of the data, or 10% of the data, and see how well a model works on the data we did not test it, which gives us a good idea how much it works in the real world, because we didn't make the model fit all the data. If it worked in the real world, it should work on data that it doesn't, we didn't train the model on. And all these techniques are techniques that they could use to really improve the data management. But beyond improving the data in this way that I've been talking about, another advantage of these things is that they could actually, we could figure out that maybe some drugs only work well for certain groups of people. And then we could redo the whole drug trial or carry over some data with only certain groups of people, such as the drug only works well in older people. So then we can redo it, only keep the data for older people and and sign up more older people in the next trial and to get the drug approved for older people. Or we can figure out who gets the bad side effects. Maybe the bad side effects only appear in people that smoke regularly, so we get rid of all the smokers and we say this drug is not for smokers. So we could approve more specific drugs. And remember, almost every drug is bad for a select group of people, even if it's generally good for the general population. Aspirin increases your chance of bleeding, which is really, really bad if you have a blood clotting disorder, even though generally it's safe. And we can go over like every drug. Some drugs are more addicted to certain people, but this will be better to figure out those people that it's safe or addicted to, so you can get more drugs approved and figure out who they're good for. And that's going to be for every drug, and potentially means that we could design more personalized drugs and treatments. A lot more better way of reviewing data. We just need to move away from what we've been doing for literally hundreds of years of how we've been analyzing data to the newer techniques that computers now can do. So this has been some of my thoughts about, in general, improving drug trials. I will go over it sometimes some more stuff, and I'll link the videos to previous videos. I hope you found this interesting. Please like, it really helps the channel. Please subscribe, that helps the channel even more. And please watch some of the other videos. Comment if you feel so, that helps. And please share these videos if it really does help the channel. And if you have any questions or anything you want to talk about, just leave a comment behind. Or if you know someone, you can have them reach out to me. Thank you very much. Bye.